Hello, everyone. If you're here to learn about the basic concept of tyrosine and phenylalanine metabolism and their related disorder, you're in the right place. Today, we're going to deal about the pathway of phenylalanine and tyrosine metabolism because it is important to understand the biochemical basis and pathological manifestation of disorders such as phenylketonuria, tyrosinemia, and alkaptonuria. All of these are genetic disorders that have high medical significance. Here's the overview of the presentation today. So as you may know, tyrosine is a non-essential amino acid. That means nutritionally it is not required, but it can be replenished by the synthesis or conversion via phenylalanine. So phenylalanine is a nutritionally essential amino acids that can be converted to tyrosine by an enzyme called phenylalanine hydroxylase. This phenylalanine hydroxylase requires one cofactor, that is tetrahydrobiopterin, to for the hydroxylation reaction. This tetrahydrobiopterin is reduced to the dihydrobiopterin during the conversion of phenylalanine to tyrosine. And for its activity, again, this dihydrobiopterin is converted into tetrahydrobiopterin by an enzyme, dihydropterin reductase, which DPH or phenylalanine hydroxylase is encoded by a gene that is located in chromosome 12, and it consists of 13 exons. This is tetramer, each consisting of catalytic site, regulatory site, and subunit binding domain. This slide shows the overall fate of tyrosine and phenylalanine. Phenylalanine and tyrosine can be obtained from the diet, and those obtained from the diet can go into various pathways, such as phenylalanine and tyrosine is one of the major component of the protein that is present in our body. In addition to that, once the phenylalanine is converted into tyrosine, it can also go to other pathways such as thyroid hormone synthesis, neurotransmitter and catecholamines, melanin synthesis, and the oxidation in the liver. And the figure here in the right shows the catabolism of tyrosine, where the tyrosine first undergoes the transamination and donates it NH3 group to form the glutamate. And the tyrosine is converted into para-hydroxyphenyl pyruvate. Next, this para-hydroxyphenyl pyruvate is hydroxylated and the homogenticity is formed. And the enzyme responsible for this reaction is para-hydroxyphenyl pyruvate hydroxylase. Once the homogenticity is formed, it is converted into 4-methyl acetoacetate with the enzyme homogenticity oxidase. And this 4-methyl acetoacetate is converted into fumaryl acetoacetate and ultimately to acetoacetate and fumarate. Therefore, the phenylalanine and tyrosine are both glucogenic and ketogenic amino acids. So this tyrosine and phenylalanine is also precursor for the synthesis of dopamine and norepinephrine, and the reaction is given here. There are 4 steps. First is the hydroxylation of tyrosine by tyrosine hydroxylase. Again, this is the enzyme that requires tetrahydrobiopterin as a cofactor. Then the decarboxylation of this DOPA formed by an enzyme called dopa decarboxylase to form the dopamine. The dopamine is further converted into norepinephrine by oxidation, and the enzyme that is required for the oxidation is dopamine beta oxidase. And finally, the methyl group is added to this norepinephrine to form epinephrine. Here, for this specific reaction, the S-adenosyl methionine serves as a methyl transport group. The first step, tyrosine hydroxylase, is the rate limiting step for the synthesis of norepinephrine and epinephrine. And the other enzyme, PNMT, is also responsible for regulating the overall synthesis, and it is regulated by glucocorticoids. The tyrosine is also the precursor for the melanin, a compound that gives the color of skin and hair. The melanin can be converted into two different types, eumelanin and phenylalanine. Eumelanin is a red pigmentation, while eumelanin is a dark pigmentation. The first step for the catalysis for the conversion of tyrosine into melanin is tyrosinase. Tyrosinase converts this L-dopa, again L-dopa, to dopaquinone, and this dopaquinone is further converted into leukodopachrome, eumelanin or phenylalanine. The mutation in the gene that is responsible for the synthesis of melanin in the melanocytes will cause the albinism. And here are the other disorders that are associated with phenylalanine and tyrosine metabolism. They include phenylketonuria, that is caused by the deficiency of phenylalanine hydroxylase, tyrosinemia type 1, that is caused by fumarate acetoacetate hydroxylase, tyrosinemia type 2, that is catalyzed by tyrosine aminotransferase, neonatal tyrosinemia, that is caused by hydroxyphenylalanine pyruvate hydroxylase, and alkaptonuria is caused by the deficiency of homogeneous oxidase. With that, thank you, and if you like this channel video, then subscribe to my YouTube channel, or you can visit www.medicalbiochemist.com for similar content. Thank you.